Good morning, everyone. I'm Ruben Sakrabani from Cranfield University. I would like to just introduce myself. I'm a senior lecturer in soil chemistry at Cranfield University. I've got a research interest in application of organic amendments with a particular focus on nutrient dynamics, looking at resource efficiency to support crop production in support of sustainable agriculture. And the focus of my talk today is going to be more looking at one aspect of the interest, which is looking at phosphorus. And I would like to call or name it as novel approaches to extracting bioavailable phosphorus for crops from sewage sludge. This looks slightly different to the topics that you have in your handout, but I will also cover that bit. I'm trying to cast a slightly bigger pitch. And this is obviously in the background that obviously phosphorus is going to be a resource-limiting nutrient in coming years. So how I'm hoping that this project idea would try and engage on how we can get renewable sources of phosphorus. The innovation behind this idea is this process called micromilling, which has been formulated by Professor Sonley, who I had the pleasure of working with as part of an EU-funded project which has just completed. And this was some spin-off ideas that was generated as part of that project. And so there's more work that needs to be done, but we've got some basic data set and we need more funding to do further work. So the process of micromilling involves these beads of zirconium oxide, which is actually used to mill activated sludge. And in the process of milling, the milling process actually breaks open bacterial cell walls, which is thick and has got this material called peptidoglycans. So with the action of milling, and it ruptures the cell wall and releases the nucleic acid, which is obviously has got lots of readily available phosphorus. And in this way, we claim that we can actually get this product called a biopolymer called biopol, which we can then use to get more phosphorus from sewage sourced or even digestives, for example, and to get a precipitate, which is phosphate, calcium, and DNA, and which is high in phosphorus content. And this product will be obviously able to provide bioavailable phosphorus that is needed for crops. So our project is trying to actually look into this in much more detail, and I'm willing to get input from others who are interested in such initiatives. And also the idea of getting this, using this biopolymer to get biophos, is that we can actually tweak the phosphorus content of the effluent so that we can apply, as you know, that's obviously limitations how much phosphorus you can apply due to the salt peak indices. So in a situation where there's high salt peak index, then we need to apply lesser amount of phosphorus, and the reverse for salt deficient peak. So we can actually control the levels using this technique. So moving on more to the crops, we propose that this technique of getting this biophos can be applicable to the combinables and also the high-value crops, horticultural crops, and also the non-food crops. We are aware that at least for the horticultural high-value crops, there will be a lot of perception issues involved in using materials that's originated from sewage. So I'm glad I'm talking first and not towards lunchtime. But also with the obviously formulation of the newly established biosolids assurance scheme and the more well-established safe sludge matrix. So we can actually tweak our methods for that. But we have also done some trials not using this material, but a similar material which is sludge origin, and we found no significant difference in yield for over 6 years of field trials. So again, that is assuring. But also what I would like to get for attention is that we are also trying to focus non-fruit crops in terms of growing energy crops on landfill sites and using waste heat from landfill site to actually dry the material and pelletize it. So we are also looking at this initiative, and we've got a collaborator from industry called FCC Environment who is keen in this, and I would welcome collaboration and input from others who are willing to be part of this initiative. Thank you.
Very much. I actually have, as opposed to the billing, I'm wearing my Ozocure hat today. And we're a relatively young UK company that specialises in oxidative technologies in a range of different applications, including agri-tech and food. There are really two main technologies that we focus on. The first is ozone, Ozone is an allotrope of oxygen. It has very potent antimicrobial efficacy. It's much better than chlorine, for example. It's much better than peroxide, about 1 million times better than peroxide. We have a variety of different solutions. So we have both gaseous ozone and aqueous ozone, and we use them in different areas. So, for example, on the air-based side of things, we're working currently with one of the largest cut flower growers in the UK, up in Norfolk. And we're applying gaseous ozone to their bulb store. And they have rooms about 4 times the size of this and about twice the height, which are absolutely full of bulbs. And we've been able to use gaseous ozone at a pretty low level, actually, to knock back both the penicillium mold formation and also to reduce the level of ethylene, which obviously reduces the amount of rot in the crop. On this year's a trial on this year's data, we're looking to get about, we appear to have got about a 30% yield increase across the bulbs compared to their existing storage methods. And the flowers that are grown from them seem to be growing much better as well. We can use ozone, as I say, both in air and also in water, but if we move on to the aqueous treatments, the next generation, the next generation actually, is a technology called electrolyzed water. And this is a really interesting technological development. It uses some developments that have recently come out of the boron-doped diamond community, so into electrophysics. And it allows us basically to turn water into a very effective, a very potent antimicrobial agent. We can do this in one of 2 ways, either with pure water to create an ozonated water solution, or if we use a variety of different salts, we can create a molecule called hypochlorous acid. Hypochlorous acid is the active ingredient in bleach. It kills everything. It's the molecule that your body uses to kill microbes, and it'll kill viruses, fungi, bacteria, protozoa, mold, whatever you need to get killed. It has a really potent effect. You can get up to 6 log unit kill with a 3-second contact time. It's just about the most potent thing out there. No resistance issues. We're producing it, and we're producing it at volume in ways that are compatible with existing irrigation and spray kit. We've done an example trial. We're actually in the middle of doing a second trial at the moment with the cut flower grower. We have run some trials on plant growth under severe pathogen pressure from Fusarium oxysporum, and we've managed to get a 3x viable biomass increase in his crop under this very high pathogen pressure. So it's very effective. It's obviously safe and non-polluting. Both of these technologies revert back to either water and oxygen, or water, oxygen and a little bit of salt as soon as oxidation has happened. They can be produced in situ. There is no taint. They can be used as a wash, a foliar wash, a root sterilant, or a soil sterilant. So lots of interesting technologies, very, very safe. They're actually FDA approved for clinical use, some of these solutions, so no human health issues either.
Good morning, everyone. My name is Sachin Shende. I'm co-founder and CEO of a small technology startup called Kisan Hub. We're based in Cambridge and I'm part of Cambridge Jet Business School's Accelerate program, as well as Cambridge Clean Tech and Future Business Accelerate Growth program. So Kisan Hub is a big data and decision support platform for agriculture. We firmly believe that growing crops is a complex ecosystem. It has got these global connections. So what happens in Australia and US matters to farmers in UK, and what happens to UK matters to farmers elsewhere as well. And at the same time, increasingly in future, farmers are going to look at disparate data sources to take the decision, or to help them in a decision-making process. So Kisan Hub is a platform where we have set an ambitious goal to curate every single key piece of data which farmer will need to make, so that they can take the informed decisions. And this includes private and public data, bringing in on one single platform. So when I say private data, it means like farmers' own data about their farms, about the crops, soil, precision agriculture data. And public data includes market, weather information, regional national statistics, data from Twitter network. So you'll be amazed to see how many farmers are using Twitter, and they regularly organize chat discussions on solving their own problems. So all of that coming onto one platform, and when farmer goes and logs in their information, they get this intelligent dashboard where farmer doesn't go to the data, but data comes to them in a very meaningful way. And all of the data is converted into their agronomic or business decision. And that's really like our aim, to look at all of the data and convert into some meaningful decision. So currently we just released our second version a couple of weeks back, which has got basic functionality in terms of market data, weather information, and functionality to so that farmers can create their own data sets and have that intelligent dashboard where the alerts and other information will appear on the dashboard. So why am I here? We strongly believe in academic partnerships and tie-ups with research firms, because we want to evolve Kisan Hub as an ecosystem. When I say ecosystem, it's like a app store type of ecosystem where researchers can come and plug in their model, where we provide all the data sets required by them and the platform to run those models. The benefit of it is really, they can take those models straight to the farmers. So it could be a pest or disease forecasting model, it could be a model in commodity analysis, it could be anything which a farmer can use. At the moment, our focus is on arable farming, but over time, we'll include livestock as well. But that's probably like down the line 2 or 3 years later. So that is one reason I'm here. Other is like really looking at similar industrial partners and see if we have synergies there, tie-up, and if we can take this product to farmers. Thank you.
Thank you. My name is Michael Lee, as you said, from the University of Bristol. I'm based at the vet school down in Langford. My background is I'm a ruminant nutritionist with a particular interest in pasture-based systems. And I really wanted to talk today about, introduce the Food Security and Land Research Alliance and also the Northwick Farm Platform. The FSLRA was brought together as a strategic alliance across the University of Exeter, University of Bristol, University of Bath, and Rothamsted Research, and in fact Cardiff University has joined this alliance. And really this strategic alliance was brought together to develop and produce solutions for the big challenges of food security. It allows us to link social scientists, animal scientists, veterinarians, economists, and microbiologists to work together to really address some of these major themes. I was really excited with my appointment at the University of Bristol because it allowed me to work with Rothamsted scientists at the Northwick Farm Platform. And I want to talk a little bit about that now today, what this is and what this can add to the agritech strategy. Agritech, and a large part of agritech, is about farming systems and how we can increase sustainable intensification of these farming systems. The Northwick Farm Platform is perfectly positioned to address and be used to help develop sustainable intensifications. So what is the Northwick Farm Platform? Well, this is a national capability set up by BBSRC, which is looking at management systems for pasture-based beef and sheep farms. So it is looking at a farm-scale system. So the farm itself has been split into 3 farmlets, each run at a different level of management. 2 of these are control systems. So one is based on permanent pasture and the other is based on increasing use of legumes. Then we have a 3rd farmlet which is based on new innovations. So that at the moment, the first innovation we're looking at on the farm platform is to have a look at the new variety of Festulonium grass, which has been developed at IBERS, which is deep rooting. So we want to have a look at the impact that has on drought resistance, but also water logging, which is extremely apt at the moment. So what we can also do with the Northwick Farm platform, we have other smaller scale catchments which allow us to investigate new interventions, which can then potentially hopefully move on to the large scale farm platform to be tested at a farm system. So what is really innovative, apart from that word, about the farm platform is the fact that we can measure all inputs and outputs, produce a full LCA approach at the farm scale level. Because each of these farmlets are hydrologically isolated. So every drop of water that falls on that land is collected in flumes and can be measured for different nutrients to produce a full NPK C cycle, so we know exactly what's happened to nutrients. So we can look at different innovations for their impact on and moving towards sustainable intensification. So some of the ideas and different projects which have been developed are listed here. A real exciting advancement is the bringing in of my colleagues at the University of Bristol Vet School and also the meat scientists. Because what this allows us to do now is to take the whole system all the way through, from the animals grazing all the way through to meat quality. We have research there actually on the quality of the meat, the shelf life of that meat, and the impact it will have on consumers, because we can also then link across to human health scientists in terms of the medics at the University of Bristol. So really all I want to do today was to make you aware of the Northwick Farm platform. If you want to do research at the farm level system, this is where you want to do it. Thank you.
Okay. Morning everybody, my name is David Booty. I'm a product development manager for a company called Omex. And I guess we stand very much at the business end of this process. So what we can offer is helping to develop practical applications for some of the new ideas that some of you have, and for developing markets too. Sorry. There we go. Who are Omex? Well, we're a UK-based independent fertiliser manufacturer, but we also manufacture in Winnipeg in Canada, in California in the United States, and we export nutrient products to about 65 countries around the world, including all the exciting emerging markets like India, Brazil, China, and other places in the Far East. So in the UK, we manufacture suspension fertilisers, solution fertilisers for arable and vegetable and horticultural crops. And internationally, as well as in the UK, foliar nutrients, so materials that are sprayed onto the leaves of growing crops. Biostimulants, things that can alter a plant's physiology to the benefit of that growing crop. And increasingly getting interested in biological pesticides, and also things like technologies like elicitors that can help to improve plants' own defence mechanisms, so improving plant health. And fertiliser additives, because sustainability in fertilisers is a real challenge, both because of pollution and the availability of raw ingredients for the future. The sort of areas that we're involved in, strategic opportunities where we're already doing research or have development projects, increasing crop yields, obviously. That's the challenge for the world, to try and feed a growing population. Trying to improve nutrient use efficiency, because what we do at the moment is unfortunately inherently wasteful. And trying not to eliminate pesticide usage, but minimise it by using other means to improve crop health or tolerance. And then the second category there, looking at things like abiotic stress on plants, so trying to get plants to grow with less water, recovering nutrients from wastewater, getting plants to deal with growing in adverse conditions where it's too hot, too dry, too wet, too cold, too salty, whatever it might be. And also another area that's increasingly of interest is the growth of biomass for anaerobic digestion. So both growing those crops efficiently to make sure you get the best feedstock for anaerobic digestion, but also optimising that process by getting the balance within those digesters right, so that the microbes do their job more efficiently. So that's who we are and who we do, what we do, and pleased to talk to any of you about any possibilities there. Thank you.
Okay, thank you very much. I'm here today on behalf of a team of us from the University of Leeds. It's a very interdisciplinary team, and my colleagues who aren't here today are Alison Baker and Andrew Cumming from the Plant Sciences School at the university, Alan Berry, who's a molecular biologist, and as you can see, I'm the chemist from the team. And we're really, what I wanted to tell you about today is a little bit of fundamental science that we've been building over the last 4 or 5 years, which we think now is starting to form a layer of sort of combined technologies and capabilities, which we think is getting to the point where it's ripe for development. And what we've been interested in is really taking an interdisciplinary approach to understanding the peroxisome and how that works in plants and actually manipulating it. So the peroxisome is an intracellular organelle found in most eukaryotes. This is a picture of peroxisomes in Arabidopsis root expressing GFP. If you can't see it at the back, these are little green dots. They're about the same size as mitochondria. It's involved in fatty acid metabolism and some other essential functions as well. And proteins that have to go into the peroxisome are targeted there by one of 2 different mechanisms, which are starting to be fairly well understood. The first thing I'm going to tell you about is a little bit of our chemical manipulation of these trafficking pathways into peroxisomes. So we've got the technology to be able to study this and screen for compounds that affect this essential pathway, and of course as chemists we can actually go and make different molecules as well. So the molecule on the right here completely meshes up peroxisomal protein import, so you may be able to see the little dots go into just cytosolic fluorescence here. Whereas this molecule here actually prevents the peroxisome from metabolizing lipids. So those of you can see, we've got here a zoomed in image of plants treated with this. They can't metabolize lipids, so these red lipid bodies are surrounded by green peroxisomes, but you can't actually use its lipid reserves. The second aspect that we've actually been getting involved with is trying to think how we can use the peroxisome as perhaps a platform for biotechnology. The reason we think this is useful is maybe we could use this organelle for storage of proteins. Why do we think this? Well, targeting signals to the peroxisome are very short peptide tags, just tripeptides at the C-terminus, which we can assess their effectivity for their import in vitro very quickly. And you can get very high protein accumulation in the peroxisome. So this EM image of a peroxisome here, this square is the protein catalase. It's been imported to the peroxisome at such high concentrations, it's actually crystallized in the middle of the peroxisome. And proteins are also imported fully folded or multimerically by the import machinery. And because proteins are expressed in the cytosol, they're not glycosylated either, which could have some significant advantages. We're not just limited to Arabidopsis, we're working now in moss, so this is the moss Viscomitrella, and we've got mutants which express or a phenotype with giant peroxisomes. So this is the normal moss here with little dots in it, and the mutant lines that we've got, as you can see, you've got these giant peroxisomes, which we think could be really interesting, as I say, as a storage organelle. So it is very fundamental science at the moment, but we're really looking for opportunities to be able to exploit this expertise moving forward. Thank you.
Okay, thank you very much for that introduction. Let me just tell you a little bit about Germain Seed Technology. Many of you probably don't know who we are. We're a company that dates back to 1871 when we were founded in California. However, through a variety of different takeovers, mergers and history, we're now based in the UK and now part of the Associated British Foods as our main owner. We are one of the largest technology providers for the seed treatment business in the world. We do not produce seeds in our own right. We don't breed seed. We basically treat seed for our customers. And we have a variety of different platforms that we use for that, which I'll describe in a minute. We're based largely in Europe and the US. A large part of our business is in sugar beet, and that's fairly historical, and that comes out of some of the technology that was developed in Germain's back in the 1940s around seed pelleting. So we are the original company that manufactured seed pellets, which some of you may be familiar with, and it's one of our main targets, one of our main opportunities for delivery of materials today. We've been involved in sugar beet production since the 1950s, and one of the reasons why we're now located and headquartered in the UK as part of our collaboration through ABF and AB Sugar as our immediate owners. We have several core technologies. Two of these, priming and pelleting, are our main ways of delivering both physiological and also physical treatments to seeds. But we're also involved in film coating, another methodology for applying active ingredients and treatments to seeds, and also involved in seed health. So that's the purification of seed to improve seed quality. Our motto, we maximise nature's potential, is basically we want to take every seed that we get, that we treat through our various treatments, and give it the potential to yield a final product and yield a useful plant as the end product. In terms of our expertises and potential for collaborations, we believe we are a scientific leader in the area of seed treatments. So certainly seed physiology, looking at improving emergence speeds, germination rates. We also look at improving storage capabilities and also cleanliness of seeds through disinfection processes that we have. We're heavily into biochemistry, molecular biology and microbiology. We have assets within our organisation looking at PCR, qPCR. We have availability of LCMS as part of the technology that we use in our everyday work. But we're looking to develop new technologies, new methodologies for rapid identification of biomarkers within seeds, look at our processes, how those processes can be improved by identifying markers for what our processes actually do to seeds. So looking for collaborative work on developing these new assays to give us some rapid technologies there. We also believe we're an innovative technology developer. We have in the past been the leader in developing pelleted technology, which most people will be familiar with, but we see this as an ideal platform for delivery of active ingredients and also for ingredients that will help plants in their early stages of development. So looking to develop those with collaborative partners as we go forward. So looking at inclusion of materials such as biostimulants, bionutrients, anything that will help the plant in its early stages of growth and improve its vigour, and obviously encourage it to reach its maximum potential. We're also, in terms of leading edge solutions, looking at seed priming and pelleting. Our seed priming technology is one of the most advanced in the world, we believe, and it gives the crop a chance to emerge very, very quickly and get established very quickly as well. But we're also looking at mechanisms which we can apply to the seed or apply in pellets, which will maybe trigger release or control or functionality within those pellets to actually create something more than just a physical transfer mechanism for the seed itself. Looking to trigger release things like bionutrients, stimulants, and also some of the outer chemicals that we also apply to seeds to improve the chances of succeeding in the real world. So that's Jermaine's. Hopefully we'll have a chance to talk to a lot of you as we go through the day. And if you've got interest, please come and see us. Thank you.
Hello, thank you for taking the time to listen to me today. I'm Dave McCutcheon. I'm the Strategic Account Manager from LGC. We're an international life sciences measurement and testing company. We have our head office in Teddington in southwest London. We operate in over 22 countries around the world and employ over 2,000 people. We provide reference materials, genomic solutions, and analytical testing products and services to customers in agriculture, biotech, food, environmental, and to the government. Key areas I'm going to cover today briefly are analytical testing, genomic solutions, reference and PT schemes, and the National Measurement Institute. With regard to the analytical side, we've been analytical testing since 1842, so we've got a little bit of experience behind us. We've been focusing in the last few years on food and ag chem, specifically residue testing, and our ISO 17025 and a GLP accredited facility. We cover a range of analytical services ranging from method development, independent laboratory validations, to crop studies, residue studies, and to routine nutritional chemical testing of products, quality control analysis, and due diligence testing. We cover a range of matrices, everything from soils, foods, waters, to crop analysis. Within the genomics side, our genomics products and services are used for marker-assisted selection and breeding, and genomic research and disease testing. We offer nucleic acid extraction, DNA sequencing and genotyping, as well as a range of genotyping laboratory automated solutions and reagents and consumables. Our goal is the development of tools and technologies to enable genomic solutions both for our customers and in our own internal laboratories. Standards division is the LGC supplies reference materials and PT schemes to support measurements, accuracy and quality control throughout laboratories throughout the world. We have over 56,000 reference materials, all which are available through our web shop on our lgc.com. The National Measurements Institute is LGC is the UK's designated leading measurement institute for chemical and bioanalytical measurement. We have a worldwide reputation in the delivery of high quality measurement science, producing robust and reliable methods and standards that underpin innovation. We look to assist in offering reliable results through development of high accurate methodology development. We manage quality assurance to ensure methods are accurate and reliable, with the provision of certified reference materials. We review and look at optimising laboratory processes and enhancing scientific skills and the development of methods throughout the UK and the world. And we ensure that we're looking at measurements as best practice. What we're looking for is strategic partners both within scientific and research organisations, within food and agri, the agrochemical and seed manufacturers, innovation and design ideas and new technology, and working in development and innovation of new technology within consultants and also within the joint laboratory partnerships. I'm David McCutcheon, and if you've got any questions, please feel free to ask me at the end. Thank you very much.
Good morning, ladies and gentlemen. My name's Stuart Cree, and I'm an agronomist and part owner of EcoSearch International Limited. The title of this presentation is Resistant Aphid Controlled Sustainably. EcoSearch International is a micro-research organisation. You could almost call us a nano-research organisation in size. Established in 1989 to develop environmentally safe products to tackle global issues. The company founder discovered the possibility of extracting bioactive compounds from arable plant material. The surprising nature previously undiscovered was that the compounds were volatile. The purpose of this presentation excuse me. The purpose of this presentation is that being micro, we do not have the resources to conduct assessments and trials at field level, and are thus seeking marketing and or research partners to test the formulation of novel aphid insecticides to tackle the EU global issue of aphid infestation and resistance to current pesticides. EcoSearch is the owner of a granted patent for the extraction and formulation of a unique plant-derived volatile compound, which is insecticidal, bactericidal and repellent. The patent was achieved without opposition from any quarter, and thus we know the molecule is unique. The molecule is extracted by a low-energy process from waste arable biomass, crops that can be UK grown and harvested by combine, so major potential for UK agriculture without affecting the food supply chain. A low carbon footprint with the biomass having compostable properties after extraction. All materials used in the extraction process are also plant-derived, offering 100% biodegradability. It is non-persistent and leaves no residue. It's possible the insecticide may have potential within the organic crop production sector to boot due to this. The extracted compound has numerous formulations, from sustainable replacement for creosote to cream and room spray insect repellents, insecticidal paints that kill any flying insects that land in it, including cockroaches, to drinking and dirty water sprays that kill mosquito larvae. We also have a formulation that can be painted on the inside of mud huts which is long-lasting and will kill any mosquito that rests there. A far more practical malaria control solution than impregnated bed nets, I might add. A further formulation can control fish lice, and yet further formulations that I can't talk about today, but which are believed to be unique globally. The point being, we know the molecule works on flying insects and water-based insect larvae, among other pests and indeed diseases. We've worked alongside independent, influential bodies and have the efficacy trials undertaken by UK academia who have confirmed our findings. To date, EcoSearch objectives have been to provide products to protect people from harmful organisms which occur in most countries and which can cause serious discomfort, irritation, disease or death. We have concentrated on parasitic transmission by insect, so virus transmission by aphids is a natural progression of the research. The question is, can we make a sustainable plant-derived aphicide formulation to control the principal aphids, pests, peach potato aphid, currant lettuce aphid by example? Is there a need? Well, aphid problems cause £12 million per annum damage in the UK potato industry alone. There are major issues with resistance build-up in aphid populations, especially with the pyrethroid insecticide, carbamates and organophosphates, particularly in central EU. Recent failure of Hallmark against the control of grain aphid causes further concern. There are no major resistances to neonicotinoids so far, but this range of chemicals have their own environmental problems, with an estimated loss to the EU economy of €4.5 billion if banned outright. Indeed, up to 90% of pesticides currently available to EU farmers are under review due to environmental issues. So what do we do? What do we need, We need someone, or a collaboration of 2 to 3 businesses perhaps, to work with us to assess our compound as an aphicide. We appreciate there are trials currently taking place on other chemicals, but these are nearing conclusion and the task of finding alternatives is ongoing. What can we offer? We are open to proposals. If organisations have the capacity to market, then exclusivity of use can be discussed. Funding of trials through academia would bring benefits to both parties and the agri-economy as a whole. Other areas beyond the EU by agreement only. I do have, by the way, an A4 summary for anybody who's interested. Thank you very much.
Hi, I'm John Willedge from the University of Greenwich. I'm part of a team that's being built up looking at micro and macro algae, seaweed or plankton in another form. Algae is probably the widest term in biology, like lipid is the widest term probably in chemistry. Algae are a very underused item, and we're particularly interested in this particular pitch in the replacement chemicals for antibiotics. Antibiotics in aquaculture and agriculture are under pressure. As you can see there, Professor Dame Sally Davis has asked for antibiotics in aquaculture and agriculture to be reduced to limit resistance. I had trouble finding really up-to-date figures in terms of antibiotic usage, but the Scottish salmon farms in 2008 used 5,500 kilos of antibiotics, according to DEFRA's figures, and in Canada they used over 21,000 kilograms of antibiotics. There is beginning to be interest in replacing these in terms of funding. The Canadian government has just put in 320,000 into the University of Alberta, looking at proteinaceous toxins to combat, to replace antibiotics, but that's a very early stage. What we do know is that algae contain a wide range of nutrients and antibiotic compounds that can have effect on the growth and immunity of aquatic organisms. One organism we're particularly interested in is a microalga called Daniella. And you'll see a lot of press releases coming out. We've got a big biorefinery project coming on looking at Daniella to produce fuel and beta-carotene, but it's been known to stimulate abalone growth. It's also known to stimulate growth in fish. They're not quite sure which compound is stimulating the growth and which compounds are stimulating the immune system. But there's tremendous potential there. We have been active in Namibia looking at microalgae, and this is a poor photo, but this is here, this yellow stain here is in a salt wash pond from salt ponds, and that is virtually pure beta-carotene. So that is a very high value product, and that's its main reason for Daniela at the moment. But we know that we can refine other products of high value, particularly of bioactivity, in aquaculture and also in livestock as well. We know that there are potential compounds that are active against, that have similar cell wall compounds to TB and to fungi. The other one that we're particularly interested in is my main funding now is in macroalgae rather than microalgae. I've grown up a bit, and so I'm in big boys algae now, macroalgae. One of the things we're looking to do is to potentially grow seaweeds with salmon farms, and then use the compounds that we can biorefine from those macroalgae to replace fungicides and other antimicrobial agents in salmon farming. So it's a big project. We have a number of funding. Have traditionally been focused in the fuels, but we know we can biorefine a tremendous number of compounds. I see the hand's gone up, and as a referee I'm not used to getting a yellow card, so I'll stop at that point. I am available if you need to ask any questions.
Hi everyone, I'm Paul Smith. I'm Head of Business Development at Incinerator Replacement Technology. Could I just wish everyone a happy Valentine's Day today? Hope you all find a bit of love in the room and start some wonderful new relationships. That's hopefully what I'll be doing today. Incinerator Replacement Technology is a bit of a mouthful. We do things with dead things. Rather than burn them, we make other things out of them. I'm going to talk to you about biomation today. Biomation is where we take a dead animal byproduct out of abattoirs, we mince it up, we sterilise it, and we cross-link that waste with an acrylic acid polymer. And what you end up with is effectively meat and bone meal in a hydrogel. Nothing new about meat and bone meal, and there's nothing new about hydrogels. However, ours is somewhat different from other hydrogels. Basically, what you can do is you can grow plants in buckets of sand, or if you like, convert the deserts into agriculture with very little water. If you look at the slides on the right, the pictures on the right there, you'll see tomatoes. This is a demonstration at NIAB at Cambridge. We've got tomatoes in sand as the top picture. After 9 days without water, and they're pretty much dead. We've got tomatoes in sand and fertiliser after 9 days, and they're pretty much close to death. And then you've got our stuff at the bottom, which is continuing to grow and thrive 9 days after irrigation's ceased. The NPK value isn't particularly high, but we do use 60% less water to achieve the results that we've got. So why is this different then? It's different because unlike most hydrogels that are used, the cross-linking of the polymer breaks down the membrane of the hydrogel, which means although it doesn't suck up a huge amount of water, it does give it up quite easily, and it also gives up the nutrient. So if you can liken the use of most hydrogels to babies' nappies, they're very good at soaking up liquid, but obviously if you've got a baby bouncing around on its backside, it doesn't release the water again. Our stuff does that. On top of that, if you're applying dressings after it's in the ground, then it soaks up the liquid dressings that you're putting in, so you get 60% less leachate from the ground, so you use less fertiliser. We think it can act as a carrier for sides, so we can embody that within the gel as well. So we think it's pretty much a major breakthrough.
We have an evaluation study going on with a big German company at the moment, and their head of technology and holistic crop care said, "You don't understand what you've discovered here." I'd like him to, well, I'd like to think it was in the context of the fact that they've got 1,400 research people at their centre, and they've been trying to break down this gel membrane for the last 10 years. He may have actually been saying, "You don't know what you're doing." Which is why we're here, because we discovered this while looking for something else completely different to kill pathogens of prions, and we want to get this to commercialisation, and we don't know what we're doing. We have no money because we spent it all on IP, so I invite anyone who's interested in working with us to put together an agri-tech catalyst grant to come talk to me and build this to move forward. Thank you.
Thank you very much. So this is just a quick overview of the position from Marks and Spencer's. Obviously, you all know Marks and Spencer's is a major retailer in the UK. But my role is as a knowledge exchange fellow. So I'm actually funded by one of the research councils, so the National Environment Research Council, to work with M&S over 2 years to help them engage with the environmental sciences and with the sciences generally. So my particular focus is on sustainable agriculture, and I've been working within their food division and their fresh produce team. So, as some of you may know, Marks & Spencer's has been leading the way in terms of sustainable retail. They launched the Plan A in 2007 with the aim to achieve 180 sustainability commitments by 2015, so one more year to go, and they're in the process of refreshing that strategy, so looking forward to the future. The commitments cross all of their business, so from stores and transport and all of those sorts of things, but also include agriculture and food manufacturing. So they require Marks & Spencer's to work with their producers and their suppliers. They have a sustainable agriculture programme which involves 10,000 farms, and innovation programmes which work with their food manufacturers. So, they have identified some major challenges, and some are on this slide, and some I've been chatting to various people in Marks & Spencer's over the last week to get a few more. So the big challenges as seen by Marks & Spencer's are around sustainable feed and animal nutrition, the whole issue of sustainable inputs, so reducing and being more efficient in the use of fertilisers, pesticides and so on. That's all linked into ideas around precision agriculture. So they're particularly interested in the ways to use information and data, but not just for information and data's sake, but how you can make that useful for performance management, for investment, for forecasting and risk management, particularly related to climate and climate change. They're also particularly interested in waste and waste management, and I'm doing a desk study at the moment looking at anaerobic digestion, because they've been talking to and have had interest from their suppliers about the role of anaerobic digestion. So these are some of the big challenges that Marks & Spencer's are interested in, and I'm here today really just to find out what's going on, what the potential might be, and to feed that back into Marks & Spencer's so that they can look at where they might be involved. Okay, so I'll be around today, so please feel free to come and have a talk to me. Thank you.
Okay, good afternoon everybody. My name is Ray Marriott. I work at Bangor University in the Biocomposite Centre, and we're a centre that really specialises in working with biomaterials, and most of those from agricultural products. The themes that we think we can most contribute to is the non-food uses of our arable crops, including biomass processing and processing of biomass-specific crops, and also the challenges in downstream food processing, and in that we would include downstream feed processing for animal feed use. The Biocomposite Centre at Bangor really focuses on biomaterials for industry of all sorts, from individual high-value molecules to biomaterials and composite boards. That was where its group originally started in the 1980s. We have bench scale to pilot scale processing facilities, and I think that's one of our USPs, that we actually, in everything we do, have not just laboratory scale, but also pilot plant and demonstration scale, so that we can produce tens or hundreds of kilograms of materials for industrial trials and formulation trials. We have a strong track record of research and commercial funding in the areas that we work in, and the areas that we are particularly interested in is extraction and fractionation of materials, using clean technologies such as carbon dioxide extraction, using liquid and supercritical CO2. And also we have a wet fractionation line which works up to 100 kilograms for processing larger and more polar molecules as well. Our experience in biocomposites, i.e. making biomaterials for boards, for packaging materials, goes back 25 years. And again, we have large-scale facilities for making up to metre by metre particle boards, for example, from novel biomass materials. To support all of this, we have advanced analysis, imaging and materials testing facilities, so we can do chemical analysis and physical testing of products. What we're really looking for in terms of partners are people that can use the facilities that we have both at laboratory scale and also at pilot plant scale. We're interested in process development from raw materials, including biomasses, through to formulating products. We offer full analytical support for any project that we would take on, and we have over 30 experienced researchers that have come from industry and from academic backgrounds, so understand the economics and practical application of some of these technologies. One of the areas that we've spent a lot of time on in the last few years is actually investing in life cycle analysis, and we have an LCA practitioner that works with us on all of the processes that we develop. So from lab scale right through to pilot scale, we are actually looking at energy consumption, raw material consumption and outputs, and actually building that into any project that we work with. I think we're really looking for partners in agricultural products and waste stream valorization, particularly in the areas of biomass, but also in the horticultural industry, where there are a number of waste streams that are generated through the processing of horticultural products, and we see opportunities to add value to some of those waste streams, or even outgrades from those products as well. And we also see applications in food and feed process developments, where we can actually take materials, even remove toxic materials by using some of these extraction techniques, and then make materials maybe more amenable for use in animal feeds. One of the areas that's already been touched on is micro and macroalgae. We've worked in both of those areas, and we see these extraction and fractionation techniques as being particularly interesting for extracting materials from microalgae and also from macroalgae. We have some captive growing systems in North Wales as well that we've been working with. So we're very much looking forward to working with partners in this part of the country, and particularly in the processing and fractionation of natural products. Thank you very much.
Yeah. The University of Leeds is one of the largest universities involved in agri-food technology, and very interdisciplinary. You've already heard some of the work this morning on chemistry that goes right through to food processing. But my own role is, I'm an ecologist who's worked on farming systems for many years. I've been very much interested in the impact of farming systems on the environment and how to measure that and how to assess that. So, for example, for my sins, I sit on ACO, which is the GM advisory panel on the environmental risk assessment of GM crops. And I'm also on the board for the Red Tractor Food Assurance, to try and get some sense of environmental processes into the whole food assurance system. What I would like to explain a little about this morning is the role of the Leeds University farm. Not too different in many ways from Northwick, for what you're hearing about this morning, but this is essentially arable. This is a commercial agricultural operation, but it's owned by the university, which means that we can try things out that aren't commercially viable at this stage, but may well be in the future. And we're particularly interested in measuring the various environmental, agronomic and economic aspects of trying things out. So what are the impacts of carbon emissions, water quality, etc., etc. And the idea is to try to link the process science, the informatics at the fine scale with what the farmers will actually do, and how that then feeds up to larger scale information sources, how that links to remote sensing, etc., to come together to actually try and test new ways of developing farming systems that are probably going to be more resilient to the present climate conditions we're facing. So the first work that we're wanting to do, which is closely associated with York, Sheffield and Ferrer, is actually enhancing the soil quality on arable farms in ways that remain cost effective but actually environmentally friendly and actually profitable. Yeah, okay, thank you.
Good morning everybody, or good afternoon even. I'm Ian Cox from the Food and Environment Research Agency, or FERA, and thanks for Les for giving half my talk beforehand. I'm a Science Bid Support Manager is my job title, and I'm joined at that by Phil Jennings. Do you want to give a little wave, Phil? He's a plant pathologist, so there are two of us here today, so please feel free to come and have a chat to us. For those of you that don't know who FERA are, we are a government agency. We're part of DEFRA. We're celebrating our 100th anniversary this year, so we're 100 years old. But I think probably more impressive is our statistics in things like the Technology Strategy Board calls. In fact, those figures up there are a bit out of date. In fact, we've won 14 competitions, including one on the first round of the Agritech Catalyst call. So it's about £4.2 million in grants we've secured over that. So if you want to come and work with the winning team, come and have a chat with us later on. We're also #1 in the UK, #3 in Europe for the EU KBBE calls as well, so we do extremely well on that side. And we do supply a lot of practical solutions to solve real world problems for our customers, whether they're government, industry, non-government organisations, both in the UK and overseas. We do have a huge science capability at FERA. As it sort of says there, bioanalytical chemistry, molecular biology, bioinformatics, modelling, risk analysis, omics, and also sort of say entomologists as well. We have a huge number of entomologists. So the chap that before that wanted to talk about his use of pesticides and that, come and have a chat with us. We can help you on that side. We're also a national reference laboratory for a variety of food contaminants, packaging, wines, you name it on that side. We're also International Seed Testing Association accredited, National Plant Protection Organisation accredited, and GLP accredited as well, for those that are interested. And so, yeah, we are open for business. Various rumours are circulating about us, but we are actually open for business. We like to collaborate with people along the way. Right, how do we deliver? We have 5 operational programmes that sort of deliver through a variety of different means into the marketplace out there. So over here we've got the Centre for Chemical Safety and Stewardship. We do a lot of work on environmental risk and fatal chemicals out in the environment there. Detection and Surveillance Technologies, this one here. It's a centre of excellence for diagnostics and analytical science and a global leader in the identification of new and previously uncharacterised threats. Plant Clinic, third one in. This is the largest plant clinic in the UK, handling something like 85,000 samples a year from a variety of commercial growers out there. Agritech Research. This is a large team of multidisciplinary science providing applied research for commercial and government customers, including developing biopesticides and sustainable protein sources. So we've got various EU projects and TSB calls on maggot farming, for want of a better word, looking at sustainable protein production out there. We have the inspectorates, bees, plants and varieties and seeds. They're based down, the inspectorates are all over the country. The bees are based up at Sandhutton in York, and the plants, varieties and seeds are based over in Cambridge. There's like the intellectual property office for plant growers on that side. We have a big food quality and safety programme out there, sort of delivering a lot of sort of analytical chemistry, including mycotoxins work both in the UK and overseas. Knowledge products and training, we do a lot of work around developing sort of new information databases and applications to get out to farmers and to growers out there. Proficiency testing, like LGC as well, we have a very large sort of proficiency testing capability to help laboratories, and also at the far end there, the Government Decontamination Service, the little yellow card there. So how do we deliver? We deliver through the Agri-Food Resilience Initiative, which is an initiative with York University focusing on food and non-food crop production systems to provide integrated approaches to look at security and environmental sustainability. The Centre for Crop Health out there, which is going to deliver sort of improvements in yield and quality for both arable and horticultural crops. And lastly, really, the Agri-Tech Catalyst, which is the purpose of being here today. So we're very happy to have a chat, both Phil and myself. So do come to site at lunchtime or later on in the afternoon. And thank you very much for listening to us.
So, yeah, I'm Carlos de Luna, coming from Whittle College, which is a specialist higher education institution in the land-based sector, located in Chelmsford in Essex. We have over 120 years of experience developing leaders in the land-based sector. Why I'm coming here is to make you aware of our infrastructure, the type of research projects that we can do here, and also to talk about our intellectual capabilities. Okay, so we have specialist laboratories that we can do nutrition, molecular genetics, we can do microbiology, we can do behavioral trials, health and welfare projects. We have a farm as well that is owned by the college that could be used for trials, that we could use for projects. We have a 90 sow pig unit. We also have arable land, around 180 hectares for crops. We have 150 sheep. We also have beef cattle. We have also access to dairy farms around Essex, especially to develop several projects that we are, especially in the early stages, some of them have evolved now into going more into the industrial side. And we have earlier this morning we talked about using technology to measure not invasively health and welfare. This is one of our projects that we've been broadcasted in the news as well, which is using satellite technology to track cows to detect, as an early system, to detect health and welfare projects. Same thing using ice tags, using thermometers to detect lameness in cattle and in other species. Another one, this project that Fairwell was featured in Countryfile, BBC Countryfile, which is the use of essential oils to modify rumen flora and try to change the chemical composition, especially of meat, so that it has less saturated fats. The same principle used in meat packaging to try to extend life, to use as an antimicrobial, as a pouch. The packaging of meat already has pouch to absorb humidity. The same principle, just to try to kill some bacteria so that it can increase the shelf life of these products. Something that I've been personally involved in trying to improve pig breeds, to trying to blend certain traits for commercial breeds and rare breeds, to try to bring the traits that are important in rare breeds that have been forgotten, trying to increase the taste. This morning we're talking about the food, the taste value of food, especially pork. So we're trying to do that, trying to bring those genetic traits. Also, we are interested in developing new ideas on how to use sustainable arable land, try to use it for ecosystem services, to develop ecological solutions to farming and to industry like you. So why are we here? I'm here to form industrial leagues to try to develop, make them aware we have these facilities. So I will be around if you want to chat with me. Okay, thank you very much.
Hello everyone, I'm Apostolos Papadopoulos, Managing Director and Owner of Crop Intellect. Crop Intellect was established in 2011. It's a micro business, and our focus is on research and development. My website actually. Yeah, our focus is research and development for developing innovative technologies for agriculture to make it more sustainable and productive. The company is separated into services, research and development, and also products. The services include bespoke efficacy trials, new product development, agronomic advice, technical support, and also support on writing bids. And our products, R&D actually is the main focus of the company, and we have won a few or several bids which allowed us to develop these technologies. These technologies are available as additives. Some of them are patented and some patent pending. And we also have products that contain these technologies. The reason for the service and the products are to bring money into the company so we can invest more into research and development, and with the aim again to develop IP and commercialize it through the large organizations that have the vehicles to take it to the market. Yes, part of the innovations and projects that we developed, and some of them are currently running, some of them came to the end, so we're applying for the next stage of development. There's a bionematicide that we developed for potato cyst nematode. We have several plans that we're looking at for compound discovery to improve the nutrient efficiency uptake for crops. We also look at extracting values from fresh produced waste, and also the way of biologically controlling blackgrass, which is a huge issue. We are based at Lincoln University, and we have a very close relationship and a lease where we can utilize the facilities in terms of the greenhouses, the fields, and analytical work. And it's part of our services to provide efficacy trials for products, and as a bespoke efficacy service, we have many years of experience in doing so, and we run these to GMP standards, and we're working really close with the customer to make sure that what needs to be delivered is properly done. So we're here today because we would like to take some of our innovations further, both to commercialize and as well closer to a commercial product, but also to offer our services, our experience and support to help you perform research, efficacy trials, and be partners. Thank you very much.
Good afternoon, ladies and gentlemen. We've had a few people this morning, this afternoon, talking about the increasing regulation and the restriction on the use of agrochemicals, specifically pesticides and insecticides. This is the space in which we're looking to work with partners, specifically to develop solutions that are alternatives or work in synergy with the existing products that are coming under restrictions. The partners that we're looking to work with are ideally experts in the area of biorationales. It's not our area, it's expertise. Our area of expertise is in production of organisms. We're looking for partners with good proven organisms for use as biofungicides, bactericides, insecticides, and growth promoters. We're also looking at post-harvest storage protection as well. We have already applied our technology to the range of organisms that's highlighted here. That's bacteria, actinomycetes, yeast, fungi production, and also we have an expertise in formulation design to ensure that the method of application, the rate of sustainable effectiveness and effectiveness itself from a chemical perspective is maintained. We're looking to develop products for commercialization, both in agriculture and horticulture. For all of you that are in this industry know that a lot of the biocontrol agents are actually used more in horticulture than arable crop manufacture. We're looking to change that with our technology. One of the problems of applying biocontrol agents is often the cost of production. We have some advanced technology that brings down the cost of production, but also added to that, there are some additional innovative features, which is that we can produce specific propagules to a very high quality standard, which looking at the industry in general, we feel that there are some significant gaps there. In terms of our technological capabilities, we can bulk up a range of organisms, and just to give you a little teaser, there's the obvious trichodermas, fusariums, glycladiums. We've also looked at metarhizium, and all these organisms so far. We've also looked at streptomycetes, by the way. So all these organisms fall within the scope of the technologies that we are offering, and we've had some very good results in terms of spore concentration, spore quality, and being able to control the specific spore, if that's what is required. Again, I just restate that we can reduce the cost of manufacture to make the use of these agents more commercially viable in large-scale farming rather than just high-value horticulture industry. The other offering we have is that if you have some specific issues with the value of the substrates making your organisms uneconomic to manufacture, then you'll find that our technology may come in very useful in reducing those costs and making it more viable. And just on the orange card, I finished. Thank you.
Okay, thank you very much. I'll move straight on to the next slide. That's slightly provocative title there, just to get everyone on. Yeah. So just before I start, certainly on the crop tables, there's a leaflet which really explains a lot more about us and introduces the contacts and so on and so forth. So I can waste a little less time on going through some of the details. What we're about is essentially a group which for many years has integrated what we think is very good physiology, especially leaf physiology in relation to photosynthesis and photosynthetic performance and interaction with the environment, and marrying that with downstream advanced genetic approaches. And we've kept pace with that in relation to current developments with genomics and next generation sequencing platforms and so on and so forth, and computational biology. But whatever we do, we're always guided by the initial physiology. And I think that has led us to many genes that we think are novel and different, and we've certainly got intellectual property on. This is one example here. But what that gives us is a whole set of different technologies which I think are more widely applicable. I should add, by the way, that our funding is primarily from BBSRC, EU, we've got an involvement with FERA, for example, in one project, and also private states funding and also Gulf state funding. It means actually, for various ironic reasons, we've got very little contact with SMEs in the UK. And this is one of the reasons I'm here, is to try and remedy that situation a little. So what we have here is techniques and technologies which I think could be useful to an audience like this. So for example, what we do effectively, you can break it down, is monitoring crop performance and health, and for using that we've developed lots of advanced imaging techniques. This panel here shows a little bit of history here. That is this last year, this is the first image of water use efficiency in a plant. Nobody's been able to image water use efficiency. We can now do it. And that is there. That means we can now screen by various techniques, germplasm, mutants, and so on, and try to then recover genes which might be implicated in improved water use efficiency, and relating always that to yield, so that we can combine water use efficiency and yield. That is not an easy thing to do, but we can now do it. Because of our techniques in molecular biology and so on, we're now in a position where we've taken approaches such as systems biology, and that has given us some very advanced modelling techniques through BBSRC funding, which has actually increased our success rate of identifying genes of importance, and we can prove importance phenotypically, at about 40% of all the genes we look at. That's a massive increase, to the point where we're sitting on more genes than we can possibly process, and we need intellect. So therefore, we've not reported them. And so we're sitting on a lot of things that could be of use and interest to people in this audience. So anyway, so we could do various things, genome-wide screening. We have various technologies to manipulate plant growth, both in terms of the physical environment, light environment, the water environment, so on, but also gene manipulation, looking at variants, and also screening by different methodologies, everything from GM to very simple mutant screening and screening of varieties. So we've got various methods for various types of performance-associated traits associated with water use, tolerance to heat, resistance to infection and so on, which we think are applicable in a wide range of areas, including things like biomarkers as well. We've got some very novel protein biomarkers which we think could be rapidly converted into kits and things like that. But I just thought I'd just pitch something that actually unites all of those things, because there is quite a movement within Europe, certainly, towards the development of pipelines of genotype of crops, which you survey and then put through a pipeline of survey to recover the genes and hopefully material. But we think, and I'm happy to talk to people about this, that we can do this now in a very cost-effective way, which really delivers and can bring in these type of developmental pipelines for even crops, even industries which probably traditionally wouldn't have thought they could access such things, such as ornamental and amenity horticulture, as well as perhaps the lower value horticultural crops, whereas most of the public investment is going to arable crop. And I think we can do this by the right business plan, going through a very bespoke novel screening systems, getting very cheap genome sequencing, but it's getting cheaper all the time, to develop intellectual property and various materials which could be used to produce the lead material. So we're very happy to talk to people, and I've got a list of people I've been listening to that I'm going to approach about talking to you about various things. So thank you for your attention.
Good morning, or sort of good afternoon, everybody. John Giles, I'm the Divisional Director at Promar International. We're very pleased to be here today, and obviously we'd like to talk to as many people as possible. I certainly won't be the first away, but I might be the last, possibly the next one. What do we do? We're a market research and consultancy company. We specialise in the agricultural and food sector. We have about 100 people work for us in the UK, so it makes us one of the largest companies of our type here. And we have three sort of key customer areas. One is working with individual farmers, largely in the dairy, but also other livestock sectors. We work for sort of trade associations and levy boards, and we also work for private agribusiness companies as well. And I'll explain it on the next slide, a little bit, the key services that we provide to them. We are in turn owned by a company called Genus, which is the world's largest livestock breeding and genetics company. They're quoted on the London Stock Exchange. Sometimes in the top 100, sometimes they're just outside. Like lots of companies, we see the UK, still have plenty of opportunities in the UK, plenty of opportunities in Western Europe still, but if you look at our website, you'll see that sort of places like China, India, Russia, Latin America, Middle East, parts of Africa have huge attraction and opportunity for us as well. Last year at Promar we worked, again, so we're UK based as well. Our head office is up in Cheshire, but we worked in about 40 countries last year. And that took us all, again, through Southeast Asia, Russia, Middle East, parts of Eastern Europe, into Latin America and North America as well. So our parent company, again, their main business is livestock breeding, so that will take them into the dairy, beef and pork sectors in particular. And in the course of any one year, we might be interacting, or our farm-based colleagues might be interacting with about 15,000 farmers in the UK at any one time. In terms of the expertise that we provide, there's three core parts of our business. One is a farm-based consultancy business. So in the UK we have about 70 people today will be out on individual dairy and other livestock farms, and they'll be providing a range of technical support to those farmers, but also business support as well. So we're very heavily involved in benchmarking, both cost of production for farmers, but also looking at things like carbon footprints as well. And we have a service called Farm Business Accounts, which provides farmers with just that. We still find that plenty of farmers around the UK perhaps don't really have a strong understanding of their cost of production. I'm responsible primarily for our agri-food work, which takes us sort of away from farms and into other parts of the supply chain. So we might be working with processing companies, retailers, food service, catering companies, and also doing some consumer work as well. Some of that work might be very local, so we might be looking at what's the demand for local foods in the Peterborough region, or Lincolnshire, or Cheshire, or South West England. At a other scale, we might be looking at what's happening in food markets on the other side of the world at a global level. And our work, again, takes us all the way through the supply chain from farm through to fork. And then we have an environmental consultancy, which is an increasingly important part of our business, which is looking at issues like how can farmers and food companies do better with regards to issues such as water and waste, greenhouse gases, carbon footprinting, environmental land management, and so on and so forth. Why are we here today? I suppose we want to find out more about the Agri-Tech Catalyst funding and strategy. We've heard lots about it. We've heard one or two presentations. I think we're still trying to work out how we fit in. We have an ambitious growth plan at Promar for the next sort of 5 years. We know that probably we can't do that all ourselves. It would be nice if we could, but we're pretty sure we can't. So we need partners to help us deliver that. And we have a long track record of working with people both in the academic and commercial sector in partnership to try and sort of mutual benefit win-win type of situations. We've just in the third year of a 3-year KTP project at Reading University, which has been very successful. Whether we go through with a formal KTP again with somebody else, we're not quite sure. We're still thinking about it, but we are committed to the principle of we can't achieve what we want to do or a task to do by our parent company all on our own. We will need partners to help do that, and we're open to talk. We're open to talk to anybody, basically. Don't feel shy. Thanks very much.
Good afternoon, ladies and gentlemen. Simon Leeds from British Sugar. I'm here to talk about, it's slightly confusing, but the AB Sugar Centres of Excellence. So, AB Sugar, and we've heard earlier about ABF, Associated British Foods. AB Sugar is part of the ABF group, so Germain's, Eric was talking earlier about Germain's. And actually ABF, as AB Sugar, it's the world's second biggest sugar company. So we're processing basically both beet and cane, and you can see beet area is basically UK, Spain, northern China, and then cane is centred around sub-Saharan Africa, so South Africa, Swaziland, Tanzania, Mozambique, countries around that area, and then South China as well. So large geographical footprint. And the mission or the vision of the Centre of Excellence for both beet and cane, and we have two centres of excellence, one for beet, which I run, head up based in the UK as part of British Sugar, and then a colleague is running the cane one based down here in South Africa. And the vision for both of them is to deliver sustainable sugar yield through innovative growth, through innovative science and technology. What do we mean by sustainable? It means lots of things to lots of different people. As a processor, fundamentally it's about having the right volume of stuff to process at the right price, which is a price that allows us to be profitable, facing into our customers, but it also allows our suppliers to be profitable, i.e. the growers, and we need to be able to do it every year. There's no point shafting people 1 year and expecting them to come back another year, because it won't happen. So fundamentally you have to get that right. But I guess increasingly, and happening at a very, very rapid rate, is this concept of sustainability in terms of what our customers want from us. So particularly the likes of Coca-Cola, Nestle, Cadbury, Schweppes, those sort of people, increasingly, they're really racking the pressure up in terms of particularly things like water, carbon, and so we have a very rigorous program to demonstrate that we're working hard in those areas. In terms of the process and the centres of excellence, we're very much looking at stuff that's out here. It was Donald Rumsfeld said we've got knowns, known unknowns, and unknowns. So we know a lot of stuff already. And actually, if we could apply that, we would see massive improvements. So in the UK, as just an example, the average crop yield is about 75 tonnes a hectare. The best growers are doing 120, and the worst growers are down to 45. So even with known technology, we've got that huge distribution. But what we're really looking for in the Centre of Excellence, and why we're here today, is what is there that's out there that by getting the right collaboration together, identifying the right partners, we can work with those people and accelerate the route to market. Because it's no good sat in people's development labs who actually want stuff out on the farm, because that's where the value is. To you as potential partners and to us as a processor. And then clearly we're looking at stuff that is way out there. And my analogy is that when I was a lad growing up in the 1980s, I had a stack of vinyl. And if you'd said to me in 2014 you'll walk around and all your music would be on something like this, you'd have said, not a chance. So what we're really looking for is the sort of the MP3 format of sugar production. So we're focused on a number of programme areas. We're not sure what the answers are yet, but these are the areas that we believe are fundamentally important. So we have already a crop model that underpins all the activity that we do. We have it both for beet and for cane. That allows us to test bed technology and really see what's going to work. Soil health and sustainability, absolutely fundamental going forward that we have sustainable soil systems. In the past we've been very silo mentality. We need to look broad at that, look across the rotation, look at how we develop that. Water use efficiency, is bang for the buck, it's tons of biomass per tonne of water applied. How do we improve that efficiency? Particularly the likes of Coca-Cola see that as really fundamental. We see it fundamentally important going forward because, water availability is becoming more volatile, but it underpins all the other technology going forward in terms of yield growth. Crop establishment for all of our crops, fundamentally we're about capturing sunlight. If you don't establish a crop properly, be it cane or beet, you won't capture all the sunlight that's available. So we have to establish crops well. We're very active in this area, but really open to opportunities around real-time data, capture of it, interpretation and management of that data, so that we can get growers making the right decisions on time. No point in doing all of the technology, clever stuff, if you can't actually apply it onto farm. I talked about the distribution of yield. We've got that now, and that's today with known technology. So there's an opportunity there immediately. Crop storage, we grow crops. There's no point in growing crops if you actually waste some of that and you never actually have the customer. So what can we do to improve crop supply and logistics? And then finally, and very much linking with Jermaine's, is opportunities around seed technology. So those are the key programme areas, and we're really looking for, why we're here today, is looking for partners who can help us support that. What do we offer? Because we contract huge volumes with a huge number of people, we grow a lot of our own stuff anyway, both in the UK, China and Africa. We provide a rapid route to market because we've got that. We basically are looking at the whole supply chain. Thank you very much.
Thanks very much. Stuart on the crops table just described me as the CD bonus track. So I'll be very brief because I know I'm between you and lunch. Just to introduce myself and our company, cheese growers are leading edge growers and suppliers of fresh produce crops, and we supply all of the UK retailers. So if you're needing any salads to go with your romantic meal this evening, just across the footbridge there are a couple of retailers in the Queensgate shopping centre. I'm sure you can get some of our products there. Basically, to fill in a bit of detail there, lettuce, celery, radish, mushrooms, beetroot, they're all core crops to our business, and we would describe ourselves as specialist growers. So we aim to be specialists within those particular areas. We have 23 members in the Cheese Growers Group. We're predominantly farming in the UK, but we also have farms in Spain, Senegal, Czech Republic and Poland. And we also have business activities in the USA and Australia. So quite a growing business in every sense. I just wanted to share just one or two sort of research ideas. I think similar to Simon, we're very keen to keep abreast of the latest technology. The reason I'm here is again to sort of make contacts and see where we can possibly work with you to deliver some of that technology within our specific context. We have got experience working with BBSRC, HAPB, TSB, PhDs, level, but I think a whole range of sort of funding streams and organisations, and we do represent a producer organisation of SMEs as well as a larger marketing company. So getting on to specifics, I'll just run through a few quickly. This is by no means exhaustive, and I'm happy to discuss any ideas with any of you over lunchtime. Post-harvest, obviously the shelf life, the quality of our products to the end consumer is particularly important to us, and as large onion growers, we're interested in detecting quality changes in storage to predict storage potential and life of the product. In terms of precision farming, we've just been awarded a KTP project, so we're planning to look at remote sensing systems, but we're also specifically interested in monitoring plant growth responses, particularly where stress factors are resulting in suboptimal crop performance. So how might we be able to detect that and measure that, for example, moisture or nutrient stress, but also what's going on in the plant that indicates maturity and quality, and how can we link those technologies together. As a group, we farm over 11,000 hectares of the crops that I've described. So from a soil management perspective, we're very interested in gauging the potential of the land to deliver our specific products at the optimum quality. In terms of water use, we're obviously interested in optimising water use, but as a supplier, grower and supplier of high value, ready to eat, and in many cases unwashed products, food safety is a primary concern for us. So we're also interested in a better understanding of the foodborne pathogens, how they behave within our environment, our growing environment, how they distributed, why and when do they appear. We manage the risk, but we acknowledge that there are gaps in our knowledge. We are the predominant celery grower in the UK, getting very specific here. So we model growth of our crops, celery being one example, based on a heat unit forecast. But we accept that whilst that's the predominant environmental factor, other factors are playing a part as well. So we'd be keen to understand a little bit more about that. And then it was mentioned at the beginning that the call is going to be focusing on plant health. And again, as specialty crop growers, we have challenges in terms of maintaining the health of our crops, specifically a range of plant and crop diseases. So any new technologies, we're very open to exploring those and to hosting trials, for example. I haven't got any slides, but my email is in the delegate pack, which you will have access to, so keen to make contact. Thank you.